Hi, my name is Kimber Salvo, and I'm here to talk about a review I did looking at the colloidal iron distribution using a method coupling Flow Field Flow Fractionation to inductively coupled plasma mass spectrometry. Now, iron is an important essential micronutrient for phytoplankton. They need it to perform photosynthesis. Without it, they can't go through primary production. And dissolved iron is considered to be a bioavailable form for phytoplankton, and a significant portion of this dissolved iron is made up of colloids. Now, let me tell you what colloids are. They're a non-sinking small particle. They're smaller than particulate iron, which sinks out of the water column. Anything smaller than 0.2 microns essentially floats. And colloidal iron is in a size fraction between 0.003 microns to 0.02 microns. And anything smaller than colloids is truly solubilized in the water. So in order to look at iron colloids as a distribution from the smallest colloids to the largest colloids, I looked at a few papers that used a method coupling Flow Field Flow Fractionation to inductively coupled plasma mass spectrometry. So in order to do that, they need to filter out any particulates first, and then they send it through Flow Field Flow Fractionation, which size separates particles out from the smallest eluting out first to the largest eluting out last. And we can attach other online detectors to this to give us additional information, like UV absorbance as a carbon proxy, or atomic force microscopy to image the colloids themselves. And then last but not least, we attach an inductively coupled plasma mass spectrometry, which will quantify the iron within the colloids. So given this distribution, do we see a varying of concentration of iron in this colloidal distribution? Is it organic? Is it inorganic? And how do these patterns change across different environments in terms of fresh water or saltier water? And looking at 3 different environments: the Yukon River freshwater, and then the Mississippi River Delta, which measures a gradient between fresh to salty, and then a high salinity environment, Goldsmear's Fjord in Sweden. So let me direct your attention to this figure here, where we have increasing colloidal diameter on the x-axis, and then increasing iron on one y-axis, and then increasing UV absorbance, or that carbon proxy, on the other axes. And the shaded region indicates that UV absorbance, and the dark black line indicates the iron. And when we see these peaks line up, that indicates we have organic iron. So across all 3 environments, we found that we have that peak alignment where we have small in sized organic iron rich colloids when we have an influence from runoff. And interestingly enough, when we look at the study in Goldsmear's Fjord, we found using atomic force microscopy at that small size fraction, we also are able to image it to be spherical and know that it's terrigenous in origin, which differs from large colloids, which were fibular in shape, indicating that they're biogenic or produced or in situ biological production. So in conclusion, we see that similar pattern where when we have high runoff, we have small organic iron-rich colloids. However, if we look at this figure here, we do not see iron quantified on the y-axis, and that's because the high salt matrices and the high salinity environment samples clogged the ICP-MS, and so they weren't able to get sensitive enough readings to quantify iron. So moving forward with this method, scientists need to figure out how to deal with those high salt matrices so we can expand this method to oceanic environments. Thank you.